So hari ni kita masuk berkaitan dengan food-borne disease. Food-borne disease, chapter 3. Ataupun dalam bahasa Melayunya ialah penyakit bawaan makanan. Penyakit bawaan makanan. So sama seperti mana chapter 1 and chapter 2. Okay, the course learning outcome is still the same, which is from this chapter, okay, the outcome is to discuss, you are able to discuss hygiene and food safety practices concepts in halal food industries. Okay, you are able to discuss hygiene and food safety practices concepts in halal food industries. So as introduction, okay, we have discussed earlier last week. So microorganisms give sufficient evidence of their presence, whether favorable or unfavorable. Okay, whether desirable or undesirable. Whether the microorganism give benefits or harmful to our health. So these organisms been effectively utilized for food production in food industry. It has been exploited in the fermentative production, processing and preservation of many foods and beverages. Ini in terms of the benefits of microorganisms. So that's why in food microbiology we studied, okay, we learn about what is the role of microorganisms, okay, whether the effect is desirable or undesirable. In terms of unfavorable aspects, there are spoilage microorganisms that causing food producers losses millions of dollars annually. So these microorganisms grow in foods and produce undesirable flavor, okay, odor, texture and appearance. So moreover, this pathogenic bacteria causing food-borne disease, okay, food-borne disease encompass a wide range of illness, okay, a wide range of illness. So this is as introduction. So we start with the mode, or the way of this microorganism being classified. So as we discussed earlier, the spreading of microbe is called transmission. Transmission. So one of the elements in the chain of infection is mode of transmission. Okay, mode of transmission, whether through air, through water, okay, through whether directly or indirectly, okay, and also through food, okay, food. So there are living vectors, and there are living reservoirs and non-living reservoirs. Okay. So food is one of non-living reservoirs for pathogenic microbe survival until its transfer to host. Okay. So the illness caused by the biological hazard or microbiological or microorganism can be classified into 3 ways. Okay. So the first one is through, we call as infection. Okay, infection. Second is intoxication. Okay, intoxication. And then the last one is toxico-infection, meaning the combination of infection and intoxication. Okay, meaning that this microorganism have both infection ways and intoxication ways. So what is infection? Infection is microorganism itself is ingested with the food. Okay, microorganism itself is ingested with the food. The microorganism been consumed, okay, together with the food. So the food may be contaminated by feces or infected animal. Example of microbes involved in this infection include Salmonella, Listeria, monocytogenes, Campylobacter jejuni, hepatitis A virus, and rotavirus. Okay, so the first 3 is bacteria, and the other two is virus. Okay, so these types of microbes, whether bacteria or virus, okay, affect a human through infection, okay, from itself, okay, directly from the microorganism directly. The second mode or the second way is intoxication. Intoxication, meaning that this specific pathogenic bacteria grow in the food and release toxins. So compared to microbes through infection, so microbes in this group, in this intoxication mode, release toxin. Okay, release toxin into the food, subsequently consumed. Okay, so the example of microorganism involved this intoxication mode is Clostridium perfringens, Bacillus cereus, and Staphylococcus aureus. Okay, and the last one is toxico-infection. Okay, toxico-infection, which is combines of two, infection and intoxication. So it's been characterized by bacteria that are non-invasive and cause illness, and at the same time producing toxins while growing in the human intestine. Okay, compared to intoxication, they release the toxins in the food, okay, into the food, but toxico-infection produce the toxin while growing in human intestine. The example is Clostridium perfringens and Escherichia coli, okay, or we known as E. coli, okay, E. coli. I think this one is not Clostridium perfringens. Here is Clostridium botulinum. Okay, sorry. Clostridium botulinum for intoxication. Clostridium perfringens is toxico-infection. Okay, so next we'll go one by one on the this pathogenic bacteria. Okay, pathogenic bacteria which affect the human health. Okay, human health. So the first one is Salmonella. Okay, so this is the types of the figure or the picture of Salmonella. Okay, and this is the structure of Salmonella. So Salmonella is bacteria. Bacteria. So Salmonella affect the human health through infection. Through infection. So this bacteria adapt and widespread in the intestinal tracts of vertebra. Okay, vertebrates, including human, Salmonella. So it is from Enterobacteriaceae family. Okay, Enterobacteriaceae family. It is a gram-negative bacteria. So gram-negative. So gram-negative means that the membrane is thin. And then it's a prokaryote. So since it is a bacteria, it's prokaryote, meaning that it has DNA without nucleus. Okay, DNA without nucleus. And then the shape is rod shape. Okay, rod shape, or we call it as bacilli. Bacillus. Okay, rod shape. It is non-spore forming. Yes, it is non-spore forming because it's infection. And then it is facultative anaerobe. Okay, what's meant by facultative anaerobe? Meaning that it can live with or without oxygen. Okay, facultative anaerobe. So it can live with and without oxygen. And then it's motile through the use of flagella. Flagella. Ini dia punya yang belakang ni. Flagella. Jaiko. Okay, cara dia bergerak motile. So it is acquired through fecal oral transmission. Fecal oral transmission, okay, from the feces and then been consumed by human, okay, fecal oral transmission. So this is the first pathogenic, okay, bacteria involved in infection. The second one is Listeria monocytogenes, Listeria monocytogenes, or sometimes we call as Listeria. So this bacteria cause the infection of, we call as listeriosis, okay, listeriosis. This type of disease we will discuss later in subchapter food-borne disease, okay, we have subchapter food-borne disease, okay. So same as Salmonella, Listeria is also facultative anaerobic bacterium, okay. So and then it can grow and reproduce inside the host cell, okay, it can grow and reproduce inside the host cell. It is a gram-positive bacteria, so it's different compared to Salmonella. Salmonella is gram-negative. So Listeria is a gram-positive bacteria. And this Listeria monocytogenes able to grow at temperature as low as 0 degrees Celsius. Okay, it can grow at chilling temperature. Okay, chilling temperature 0 degree Celsius. Okay, the third bacteria we call as Campylobacter jejuni. Okay, Campylobacter jejuni. So the name means curved root. Okay, it is curved root. So it's a helical shape, compared to Salmonella and Listeria. They are in root shape, but this Campylobacter is in curved root shape, or helical shape. Same with Salmonella and Listeria, it is a non-spore forming, and then it's a gram-negative, okay, gram-negative bacteria, and then it is a microaerophilic, aerophilic bacteria. It is non-fermenting. Same with Salmonella. It's multirods with a single polar flagellum. It has flagellum. It has oxidase positive and grow optimally at 37 degrees Celsius or 42 degrees Celsius. This bacteria commonly associated with poultry or poultry products, including the eggs. So naturally, this bacteria or Campylobacter colonize the digestive tract of many bird species. That's why normally when we do the hazard analysis for products involve poultry, normally we put one of the biological hazard is Campylobacter jejuni. Okay. Okay, next is virus, which also affect a human through infection. So the first virus is hepatitis A virus. Hepatitis A virus. So this virus, of course, same as its name, it causes hepatitis A disease, or formerly known as infectious hepatitis. So it is an acute or short-term medical care infectious disease of the liver. So it attack the liver, liver of human. So many cases have few or no symptoms, especially in the young, where 90% of children do not have symptoms. Okay, so normally when it attacks human, especially the young or the children, they have no symptoms. Okay, so the risk for symptomatic infection is directly related to age. more than 80% of adults having symptoms compatible with acute viral hepatitis. But compared to adults, majority of children having either asymptomatic or unrecognized infection, meaning that they don't show any sign of disease. Okay, when they get the hepatitis A disease. Okay, the second virus that affect human through infection is rotavirus. Rotavirus. So this is the most common cause of severe vomiting and diarrhea among infants and young children. So a genus of double-stranded RNA virus in the family of Reoviridae. Okay. The immunity develops with each infection, so subsequent infections are less severe. Adults are rarely affected. So this virus mostly affected the children, infants and young children. So it's rarely affected adults. So there are 8 species referred to: rotavirus A, rotavirus B, C, D, E, F, G, and H. But compared to other species, rotavirus is the most common species because it causes more than 90% of rotavirus infections in human. So same with other bacteria, this virus transmit, okay, by the fecal, okay, oral, fecal oral route, okay, meaning from the daripada apa? Kita panggil sistem perkumuhan, and then tercemar masuk ke dalam makanan kemudian kita makan, okay, fecal oral route. So this is the first mode, the first mode of how the microbes affect the human health. How these microbes bring illness to human. Okay, the second mode we call as intoxication, which is these microbes release the toxin into the food. Okay, release the toxin into the food. So the first microbes that involved in intoxication mode is we call as Clostridium botulinum or C. botulinum, one of the most dangerous, okay, pathogenic bacteria, Clostridium botulinum. So it grows on food and produce toxin. So when ingested, it can cause paralysis, lumpuh. But it is extremely rare. Okay, it's rare, but it is very dangerous. That each case is considered a public health emergency. Okay, public health emergency. So this bacteria is gram-positive, same as listeria. It is a gram-positive. It is rod-shaped, same with Salmonella and Listeria. It's anaerobic, and anaerobic meaning that it can live without oxygen. So these types of bacteria mostly concern in canning process. Canning process. That's why in canning process we do the sterilization until 121 degrees Celsius to make sure this type of bacteria is dead. However, it is more dangerous if this bacteria can release the toxin before they are being dead. Okay, they are spore forming. So the spore can resist to the temperature, and then when they have a suitable temperature, they will grow again. Okay, they will grow again. Spore ni macam telur lah, telur dia. It is a multibacterium its ability to produce the neurotoxin botulinum. Okay, neurotoxin botulinum, or we call as Botox, Botox, botulinum toxin. It causes a severe flaccid paralytic disease. Dia memang boleh menyebabkan lumpuh. It's the most potent toxin known to mankind, natural or synthetic, with lethal dose of 1.3 to 2.1 nanogram per kilogram in humans. So in cosmetic, sometimes they use their toxin as material, okay, to beautify their faces, okay. But if the dose is incorrect, it will bring lethality, okay. So this is the first bacteria that affect the human through intoxication. Okay, the second bacteria we call as Staphylococcus aureus. Okay, Staphylococcus aureus. So from that name, we know how its morphology or its structure. Coccus, coccus means sphera, sphera. Staphylococcus because the shape is like grapes. Okay. So it is a gram positive. Same with E. coli, and same also with apa tu tadi listeria gram positive. So it's non-moving, small round shape. It is non-motile compared to bacteria, compared to seaboards, compared to listeria, compared to salmonella. So this type of bacteria is non-motile. Okay, non-motile. It is like grape, like grape. Staphylo, okay, staphylo macam apa? Gugusan, gugus. So it affect human through what we call as disease of staphylococcal food poisoning. Staphylococcal food poisoning. Okay. Normally, these types of bacteria is come from food workers, or human factor. For example, food workers who carry Staphylococcus aureus and then handle food without washing their hand, contaminate foods by direct contact. So it can be found in unpasteurized milk and cheese product. Okay, normally in milk and cheese products. And then these types of bacteria is salt tolerant. Salt tolerant. So we need to give heat treatment to kill this bacteria. So the salt is not affect for this bacteria. So the pH is not have any function for this bacteria. And then since Staphylococcus aureus is producing the toxin, so the toxin are resistant to heat and cannot be destroyed by cooking. The bacteria itself can be destroyed, but when it release the toxin, so the toxin can resist to heat. Okay. The bacteria number 3 with intoxication mode is a Bacillus cereus. Okay, Bacillus cereus. So we know from that name how is the shape of that bacteria. Bacillus is in rod shape. Rod shape. And this bacteria is also gram positive. Okay. It can motile. Okay, it can motile because it have a flagellum or flagella to motile or to move, mobile. It is a beta hemolytic bacterium. Okay, beta hemolytic bacterium, meaning that it has ability to induce membrane of red blood cells by hemolysis when grown on blood agar. Okay, it's have ability to hemolysis or to divide, okay, the red blood cell. Okay. Normally, this type of bacteria is known with fried rice syndrome. Okay, like its name, serious. Normally, this type of bacteria is we can found in cereal products, including the rice. Okay, rice. It is one of the most cases lah in Malaysia when relate to food poisoning in school. In school, fried rice syndrome. It is facultative anaerobe. Okay, so it can live with and without oxygen. So it can produce protective endospore. Okay, we have protective endospore spores. Okay. Okay. Next is the third mode of how the bacteria or microbes affect the human health. We call as toxico-infection. Means that it combines the infection mode and the toxication mode. So the first one is Escherichia coli, or sometimes we are familiar with E. coli. Okay, E. coli. So E. coli is gram-negative. Okay, same as Salmonella. It is gram-negative. It is facultative anaerobe. It is rod-shaped. Okay, commonly found in lower intestine of warm-blooded organisms. Okay, normally it's been released from our feces. From our feces. Mostly this type of bacteria can be found in sewerage treatment plant. Sewerage plant. Okay, kat pelang kumbahan. Loji kumbahan. So some serotypes can cause serious food poisoning in their host and are occasionally responsible for product recalls due to food contamination. Okay, however, not all E. coli is harmful. Okay, harmfulness. Some are harmless. For example, E. coli in our gastrointestinal tract system, GIT system. So it is a part of our we call as macroflora in our gut, okay, or our GIT, gastrointestinal tract. So the fecal oral transmission is the major route. Okay, it expel into the environment within fecal matter. Okay, so bacterium grows massively in fresh fecal matter under aerobic conditions for 3 days. But its numbers decline slowly afterwards. Okay, the second types of bacteria, okay, which use toxico-infection mode is Clostridium perfringens. Clostridium perfringens. So this bacteria is gram-positive. It is a rod-shaped, anaerobic. It is spore-forming pathogenic bacteria. The Clostridium perfringens enterotoxin, okay, mediating the disease is heat level inactive at 74 degrees Celsius, meaning that we need to treat our food, okay, more than 74 degrees Celsius to kill this bacteria and its toxin. It is enterotoxin. So it can be detected in contaminated food if not heated properly and feces. The incubation time is between 6 and 24 hours after ingestion of contaminated food. So how we know that there are Clostridium perfringens is our product? So the symptom can be seen, okay, after 6 hours or until 24 hours after ingestion of contaminated